Our next talk, we have Colleen Durkin from MBARI, who's going to give us a visual tour of sinking particles. Excellent. Over to you, Colleen.
All right. All right. So thank you for inviting me to talk today. I'm going to tell you about this paper that we recently published in Global Biogeochemical Cycles. And I did this work with my co-authors, Ken Bissler, Ivana Sejtanic, Meg Estapa, Pat Kelly, and Melissa Oman. And the title of our paper is A Visual Tour of Carbon Export by Sinking Particles. And so today I'm going to take you on a tour. But before I start that tour, I just want to acknowledge all the people who were involved in this work. First of all, not listed here, there were 4 reviewers of this paper. So if you were one of those reviewers, thank you very much. I appreciate your contributions. And then also all this work, we sailed on 4 different ships to do this work, and it wouldn't be possible without the captain and crew of those ships, and also the science teams that sailed. And I already mentioned my co-authors, but there were some other scientists that really contributed. And then of course our funding started with a pilot study funded by the Rhode Island Endeavor Program. NSF funded us with an eager grant to keep working on this. And then the Schmidt Ocean Institute, and we funded one of our cruises, and then that really set us up to be part of the NASA Exports program. So thanks to all of those people. So the topic of this work focuses on the biological carbon pump. And fortunately, I think this audience knows a lot about the biological carbon pump, but I'll just give you a quick overview. So it starts with when phytoplankton fix inorganic carbon into organic matter in the surface ocean through photosynthesis. And then a fraction of that carbon can sink into the mesopelagic through sinking particles, like some of these examples here. And then an even smaller fraction actually makes it into the deep ocean, where it's sequestered from the atmosphere for long periods of time. And I'm pretty sure you all know, the biological carbon pump is one of the most poorly constrained parts of the ocean's carbon cycle. And so that's due in part because of all these really complicated biological interactions involved in both fixing carbon and then transforming that into these sinking particles. And so this study really targets the biology of these sinking particles in order to try to better understand and constrain the biological carbon pump. So I told you I was going to take you on a tour. This is our tour map. These blue dots are all the locations where we collected sinking particles for this study. And then just as a teaser, in the future, there'll be more data coming from the NASA Exports field program that was at the Porcupine and Bissell Plain. So the first place we went was the New England shelf break in November of 2015 and 2016. And then that was that pilot study led by Melissa Oman. And then we sailed from Hawaii to the coast of California on the Sea to Space expedition, which was on the Falkor, and collected sinking particles at these 3 locations. And then finally, we applied all these methods that we had developed on the NASA Exports cruise in the North Pacific. And so at all of these locations, we collected sinking particles using a variety of methods. These are the different types of sediment traps that we used. So we used this really traditional design, the surface tethered trap with a surface buoy, and then an array of traps tethered below it. So here's an example. Here's a frame with some trap tubes attached at various depths. We also use neutrally buoyant sediment traps, and then this kind of unconventional design where we put a trap below the profiling part of a wire walker. So all of those trap tubes collected sinking particles. Some of those tubes had formalin at the bottom so that we could measure bulk sinking carbon. All of those arrays also had a tube with a jar containing a polyacrylamide gel at the bottom so that we could capture individual sinking particles. And so that's what this looks like. So for example, at this location in the North Pacific, this is one of those jars. So we're looking down at the jar, and you can see that the benefit of using these polyacrylamide gels is that when the particles settle into the gel, they mostly maintain their original size and shape, and so we can see, really quantify them in their thin condition that they were sinking. So after we recover the traps, I put them on the microscope and take a lot of images. First of all, I image almost the whole gel area at a low magnification, so that is really important for quantifying those rare large particles. But then you increase magnification so that you can quantify smaller and smaller particles. So this generates hundreds of images for every sample. And then I put those images through an automated image analysis process, which I wrote using tools in Python. And if you're interested, all that code is available on GitHub. And if you want to use it or improve upon it and you have some questions, please email me. I'd be pretty happy to talk to you about that. But just as in summary, basically what the code does is puts the images through a series of transformation steps, thresholding, edge detection, in order to finally detect the likely particles in the image. So just from this, you can get how many particles are there and then what the size of the particles are based on how many pixels it takes out. But we have a lot more information too, because we have a lot of biological information from the images. So at the end of that processing step, we're left with a huge file full of these cutout particle images. And the next step is to classify them into similar categories. And so, for example, here are various categories of particles that we detected, and we are only interested in the passively sinking particles. So you can see there's some zooplankton that we are pretty sure swam into and got stuck in the gel. There's also these fibers, which we haven't decided what to do with those yet. So we exclude the non-passively sinking particles. And what we're left with is these 9 different categories of sinking particles. And so we have phytoplankton, which we can resolve because of that high magnification. And there's also often a lot of rhizaria in the gels. And so here's examples of these really large phaidarians. And then we also see more traditional looking marine snow, like aggregate, which are these translucent, fluffy types of particles. Then this other type of aggregate, which I just called dense detritus, which could be fragmented fecal pellets or formed by some other process. And then a lot of different types of fecal pellets. So there's these large loose pellets, which are probably produced by zooplankton, that non-crustaceous zooplankton like pteropods. Then the long pellets, which are really typical of the crustaceans. Self-fecal pellets, which are really distinct. Mini pellets, which are, these are usually less than 50 microns, so they're really small, and they're produced by microzooplankton. And then these short pellets, these short oval pellets that are also produced by a small zooplankton. So, for example, like this football-shaped pellet is really typical of like a larvation fecal pellet. So with all this biological information, we can make intelligent choices about how to model the carbon content and the carbon flux by these particles. And to do that, we've already measured the particle size using that image processing step. And then I use that to model the particle volume, which is based on the most likely shape of that type of particle. So it's specific to each category. And then using some variation of this equation to model the carbon per particle. And so these two parameters are specific to the category of particle. And then finally, you can multiply that carbon per particle by the number of flux in order to model the carbon exported by every particle type. And so how well did we do? Here is the measurements we took at that location in the North Pacific. We collected sinking particles between 100 and 500 meters. And this is the measured carbon flux from those bulk formalin preserved traps. And this is what we predict based just on imagery. So the model does pretty well. I was really excited about this. But we can say more than this. We can also separate that modeled carbon flux into different particle types as well. And so what I'm showing you here is just one of our trap deployments, but we were out there for a whole month and did this 3 times. So this is all 3 deployments over the course of a month. And so you can see how we did each time between the measured POC flux and the models. And what you'll notice about this location is, first of all, carbon flux is really low, between 1 and 2 millimoles of carbon per meter squared per day. There's really high attenuation with depth. And there's this week-to-week variability in the composition of the particles that's largely driven by this episodic presence of salt fecal pellets. So takeaway from our first location on the tour is that these weekly variations in carbon flux were driven by the episodic presence of salt fecal pellets. And the next stop on our tour, we also took depth carbon flux measurements with depth, and that was here at the New England Shelf Break. And so I'll show you what was going on at that location. Here's the same type of graph. We went out there in November of 2015, and then we went back to the same location the next June. So what you'll notice here, we have the modeled POC flux was much higher at this location, between 15 and 20 millimoles of carbon per meter squared per day. And that was true at both months that we were there. However, they were seasonally extremely different in terms of what was causing that carbon export. So in November, it was largely composed of these diatom aggregates. This is a fluffy aggregate that's completely full of costinodiscus cells. Whereas in June, that carbon export was composed of these long crustacean fecal pellets. And the thing is they attenuated extremely rapidly with depth. And so that got us interested in thinking about what the attenuation profiles of these different particle types are. And so what I did is I took this data and combined it with that North Pacific data in order to see if there's any general patterns that we see. And so that's what you're looking at here. In order to put those 2 data sets together, I had to normalize them since they were collected at different depths. So this is the depth here is the meters below the 1% par, and the x-axis is normalized POC flux. And so what you'll note, and the different colors on these graphs are the different trap deployments. What you'll notice is those long crustacean fecal pellets attenuate extremely rapidly with depth. I calculated an average B value, which is that Martin curve attenuation with depth. The slope of that attenuation is 1.8, which is really high. Aggregates have this more average 0.8 attenuation with depth. And then the salt fecal pellets, there's no evidence of any attenuation with depth. And so what I'm showing you here are these 3 largest types of particles that we see in the traps. But if you look at the small particle types, it's also pretty interesting. So for example, here are those mini pellets that are less than 50 microns. And they also have this kind of moderate or kind of high attenuation with depth, -1. And then these short oval fecal pellets at times actually look like they're increasing with depth. And I think that makes sense because of, if we think about the source of the particles, these short pellets are, I told you that some of them are pretty typical of things like larvation. Oops, I can't control my clicking. Okay, so it makes sense that larvations might be living in the mesopelagic and repackaging particles into their fecal pellets, so that the flux of this type of pellet could increase with depth, and it's evidence of repackaging. I'm also really fascinated by these mini pellets, which are produced by heterotrophic protists, and especially I'm super fascinated by the potential of them being produced by the Phaeodarians, which also live in the mesopelagic. Takeaway from this second stop on our tour is that we saw this big shift, this seasonal change in the ecological mechanisms of carbon export at this location, even though the magnitudes out of the surface were pretty similar. And then when we think about those locations together, we saw that the attenuation with depth varies according to each particle type, and it wasn't so much determined by particle size. So the large particles can attenuate very rapidly with depth. And then, so I want to just use this last part of my talk to think about particle size and take advantage of these three last locations where we only had one trap depth deployment at 150 meters at all three of those locations. And so I'm going to show you that data separated by size. Sorry, my computer is very slow. Oh, here we go. So now what I'm showing you is that model of POC flux now that's on the y-axis, and the x-axis is the equivalent spherical diameter. And so at these two oligoterific locations, the particle sizes were all less than 1,000 microns. And you can see that when we get to particles smaller than 100 microns, they're largely composed of things like these mini pellets. And once we got to the California current, we got a shift. There was particles that were greater than 1,000 microns, and you can really see that the larger particles are there's more fecal pellets in that size category, right? Especially when we were there were a lot of these long fecal pellets. So you can see that the small particles can be important contributors to carbon export in terms of magnitude, and also that they're made of different types of particles. And so this is just to try to show you that with the importance of those with actual data. So now I just put all the samples together from all the locations on our tour to try to look at the importance of the small particles in every single sample that we collected. And so here I'm showing you the fraction of the total POC flux by less than 100 micron particles. And I plotted that against the total POC flux at that location. So what you can see is that at these locations with high carbon export, New England shelf break, there's just this really consistent contribution. About 5% of the POC flux is packaged in these less than 100 micron particles. And so it doesn't really matter. They're just consistently important, at least 5% important. And then, but once we get to these really low flux locations and samples, less than 2 millimoles of carbon per meter squared per day, the small particles start to compose a much larger fraction of the carbon export. And on average, this is 19% of the POC flux. But you can see some samples, it's almost getting up to 60%. So they're important contributors. So that's the end of our tour. And the highlights are that thinking particle imagery can reasonably model POC flux. We saw short-term variability that was driven by episodic fecal pellet fluxes, and in that case, would be felt. We also saw that we saw evidence of seasonal changes in the ecological mechanisms of carbon export, and I think just intriguing. We saw some intriguing evidence that particle type rather than size determines depth attenuation. and that small particles are important contributors to POC flux, and they often have a different ecological source from the large particles. So I hope I have time for questions, and also if you don't get a chance to ask your question, you can send me an email. Great, thank you.
Thank you very much, Colleen. Another excellent talk, more fascinating stuff to discuss. So over to the audience. So we have one already. Cordelia, would you like to ask that question rather than me risk reading it incorrectly from the chat?
Yeah, sure. That was a really great talk and really interesting. And I sort of followed this from the preprint, so it's really great to kind of see that it's come to fruition. I just wondered if you sort of early on in your slides, you talked about whether you collected some data for DNA analysis, and I just wondered if you'd started or got any results on that.
Oh, I'm so glad you asked. Yes. Yeah, so we also isolated individual particles and sequenced the DNA in those particles, and that paper I hope will be published soon. But yeah, it's I'm excited about it. Yeah.
Did you say that was at an individual particles, so single particles?
Yes. Yeah. So from that, so right now we have a paper, like a study written up with about 80 particles from just that transect between Hawaii and California. But we did that also in the exports cruises, and we actually have about 1,000 particles to analyze there. And that DNA is almost all sequenced. I've been working my butt off in the lab, and this is probably a good opportunity to say that we're looking for a postdoc to help analyze that data. So you can apply right now if you're interested.
Excellent opportunity there for someone.
Amazing, thank you.
Thank you, Cordelia. So we have a question from Jessica as well. Jessica, would you like to sort of ask your question in person?
Oh yeah, sure. Thank you. Colleen, very interesting talk. I was just wondering about this particle size and depth attenuation relationship. If you were to exclude the salps and appendicularians, do you see a relationship between size and depth attenuation?
Yeah, I didn't actually thought it that way. I think that would be worth. I don't know. I think I was thinking about it really in terms of different particle types, but I do think it'd be worth plotting it versus just particle size. Yeah, I don't know, but I think that's a good point.
Okay, thank you.
I don't want to say that size doesn't matter at all. Like, of course, smaller things are going to sink slower, but
yeah, you just have exception. I mean, basically the salps and appendicularians are somewhat exceptions to the rule. Yeah. Mm-hmm. Yeah. Thanks.
Thank you, Jessica. I can see that Pierce has his hand up. Pierce.
Hello. Thanks, Colleen. So I was on these voyages. Were there people measuring the phytoplankton community as well?
Yeah. Mm-hmm.
I just wondered whether there were any consistent relationships that jumped out to you between that community composition and then the kind of export you get, the kind of particles that are then sinking into the mesopelagic.
You mean the phytoplankton or the zooplankton?
Well, I suppose it's more directly related to the zooplankton, I suppose. But both would be useful. Yeah, just thinking off the top of my head.
Yeah. Well, what I do think is that based on this DNA analysis, whether certain phytoplankton are going to be packaged and exported, it's important which zooplankton are present. So like a large diatom have the potential to aggregate and sink on their own and be collected in on traps, whereas really, really tiny tiny phytoplankton, they need something else to package them into some kind of a particle that's going to be large enough to escape the mesopelagic or the surface. So something like the presence of salps will make things like Senecacoccus contribute more to export than if the salps weren't present.
Okay.
Yeah. I don't know if I don't think that answered your question, but
Well, no, it did, but I guess I don't want to take up too much time, but I just was just thinking, if there's a particular percentage of, say, you mentioned Senecacoccus, if Senecacoccus represented, say, a particular percentage of the phytoplankton community, then does that then support a zooplankton community that tends to generate these long fecal pellets, say, that are so important for export? That's kind of my line of thinking.
No, I think there's a lot of room for to explore that. I mean, this was kind of our first presentation of this kind of approach, and I hope that it will generate more studies trying to make those ecological linkages. So, hopefully it's going to lead to better models.
Cool. Really, really cool stuff. Thanks.
Excellent. I think we've still got time for one more question if anybody has anything out there. Let me just check the chat. I don't see anything. So as we're coming up to the time anyway, thank you very much, Colleen. That was excellent. And it's also good to see that you've put the code on GitHub. It's always nice to see, the software being made available. So hopefully you will get a lot of people making use of that.
I hope so.
Yes, I hope so.
It took a long time. It took a long time to figure out, so I hope it's useful to somebody.
It definitely deserves to be made use of. So thank you for putting that out there for the community. That's it for this month's call. Thank you once again to both of the speakers for giving us their excellent talks. Thank you to everybody for joining the call. And then just to